Hello and welcome to Chapter 14, Part 1, Gene Expression from Gene to Protein. I'm Dr. Chris Whips. Let's get started. All right, so it helps to think about, well, what is gene expression? And we can think of this like a flow of genetic information. And in gene expression, we're taking that information that's stored in DNA, that's encoded there, thinking of DNA as kind of like the library, the instructions, and then taking those instructions and going through a couple of stages called transcription and translation to ultimately express what is coded for in that DNA. And so we're going to go through transcription. That will produce a piece of RNA, which is then read in, and actually that's where the decoding takes place, and proteins are built from that. And so proteins are really that ultimate expression, usually, outward expression in a particular organism. And so this really provides us that link between the underlying genetics and then what's happening outwardly at the phenotypic level. So to understand how gene expression works, we can start with a model organism, Neurospora crassa. That's the bread mold. And these guys here, Beadle and Tatum, did research on bread molds. And one of the basic setups they used was to irradiate with X-rays their bread mold cultures and really trying to knock out certain genes. And then if you can knock out sort of systematically different genes, you can figure out what genes are doing and what they're expressing, and or when you've got a mutant, what has been knocked out, as it were. And so they have their bread mold cultures. They don't grow them on a piece of bread like I show you there, but they would grow them on a culture medium. And so here we have Neurospora cells growing on a complete medium. So this has all the nutrients these things would need. And that means they don't need to synthesize any nutrients themselves. And so even a mutant that has a certain gene knocked out should be able to grow on here, unless it's a completely other important gene that means it can't grow at all. But those aren't interesting in this case. And so what they did was, here's this irradiation, and then they grow again on the complete medium. And then they see, well, what happens when we put this in each of these irradiated cells into different media? So if we put them here into this medium here, this is called minimal medium. It's lacking some of the nutrients. And what do we see? Oh, the cells don't grow. That means this particular mutant can't synthesize the nutrient that's missing in this particular minimal medium. And so this minimal medium is missing, say, some amino acids, as in this case. So we can add back. Let's add valine to this medium. Okay, still doesn't grow. Let's add lysine. No, still doesn't grow. Let's add arginine. Oh, we've got some growth now. So it would seem that if we give this mutant arginine, it can survive, which then tells us, looking back, that some gene in its ability to synthesize arginine on its own has been knocked out. That's what's been damaged here. And we know that they can produce arginine normally, because here's our wild type cells growing on minimal media, and they can grow. That means they can produce arginine. So let's look at a little more detail at this. This arginine synthesis is a biochemical pathway. Now you have to think back a few chapters to biochemical pathways. Look at this example of a biochemical pathway. Mutation results in defective enzyme B. What would be a consequence of this mutation, this damage to enzyme B? Now, these A, B, and C are substrates that the enzymes are acting on, or products, as we see C and C. So what would happen if B was knocked out? All right, yeah, so if enzyme B is knocked out, you're not going to produce C, and that means B will likely accumulate. So by using that same logic, this is how Beadle and Tatum figured out the biosynthetic pathway for arginine. So they found 3 different mutants. Here we're going to call these gene A, B, and C. They produce enzyme A, B, and C. And they're important for converting this precursor, say, to ornithine, which is then converted by enzyme B to citrulline, which is converted by enzyme C to arginine. That's how these make arginine. And so they found by chance knocking out these different enzymes that they had different production of these compounds, and they came up with what's called their one gene, one enzyme hypothesis. So here's gene A produces enzyme A, and so on. Now let's look at their experimental results more closely, and we'll take our wild type strain of Neurospora. This has no mutation, so it grows on normal media, and even if you take arginine out of the medium, it still grows because it can make its own arginine. And even if let's add back ornithine, still grows, because it doesn't matter what step you're knocking out or supplementing, the wild type will grow. But if we knock out gene A, our wild type or our mutant here can grow on medium. This has all the amino acids these things need, so it can grow. But if you take out arginine, because these guys can't make arginine, it will not grow if we take away the arginine. But if we add ornithine here, look, it recovers the growth. Why is that? Because remember, gene A is mutated, but if we give it ornithine, it still has B and C, so it can kind of pick up from here. If you give it ornithine, sure, great, I can make arginine. And so that same logic applies here, a gene B mutant, so gene B is knocked out. If we give it ornithine, it won't matter because gene B is knocked out. Ornithine comes before enzyme B. It's the substrate for enzyme B. If we give it citrulline, of course, that's a little further along, and there we go. We can rescue that. So that's the logic behind figuring out these biochemical pathways and this one gene, one enzyme idea. So this has really evolved over time, and there's a few things that you probably already know. Not all proteins are enzymes, right? So that one gene, one enzyme hypothesis doesn't really work for everything. So this sort of morphed into this one gene, one protein idea. Now, we also know that some proteins are made out of multiple polypeptides. And so, maybe there's multiple genes that have to come together to form an enzyme or a functional protein. And so this sort of evolves into this one gene, one polypeptide hypothesis or protein product. And then we have to add another layer, this idea that sometimes just RNA is the product. It doesn't go any further than that. And so probably the best way to describe gene expression is here at the bottom. It's DNA sequence that's expressed to form a functional product, either RNA or a polypeptide. And so here's what's known as the central dogma of molecular biology, this idea that we're taking DNA, stored information as DNA, it's being transcribed, copied into a temporary piece of messenger RNA, and that message is read, and proteins are built from it. Again, sometimes we stop here just with a piece of RNA, but most of the time we're building a protein. And so this flow of information is DNA to RNA to protein. What's going on in between is transcription and translation. Those are the arrows. But the central dogma, this is one of these top 10 things to know 10 years from now. DNA to RNA to protein. We can add some little wrinkles to this if we want. Again, this is the central dogma up here, but we can recognize that, DNA is copied and replicated. So we could say, DNA can be amplified that way through replication. Some viruses can undergo reverse transcription. They can take RNA information and convert it to DNA. And so you can add a few little wrinkles here, but the bulk of what's important, especially at an intro level, is this: DNA to RNA to protein. Know it cold. So here's what's happening if we look at the cells, and we'll talk more about this. Prokaryotes, DNA to RNA to protein. But notice this, you do not have compartmentalization like you have in a eukaryote. DNA to RNA to proteins all happening right here next to each other. That'll be important. Whereas in a eukaryote, DNA to RNA, that's happening in the nucleus. And then what's happening outside in the cytoplasm or elsewhere is translation. That's where the translation's occurring to protein. Now you can think of these transitional processes, transcription and translation, almost like thinking about language. So if we say we're transcribing DNA to RNA, we can think of that almost like the same language when you're just transcribing something, you're copying it down. Because we're using the language of nucleic acid. We know that RNA is slightly different, but more or less it's the same. They're nucleic acids, and so that's why we would call this transcription. Whereas translation, we're almost taking what's in one language, nucleic acid, and translating it into amino acid speak, which is protein. So that's why we use the term translation for that. Here's a slide that you can come back to from time to time when you're thinking about these different processes, what makes them similar, what makes them different. We'll cover all of these things, but potentially important points to note now is this template, DNA to RNA, and translation is RNA to protein. And then what are we using to build these things? Well, if we're building a piece of RNA, we need RNA nucleotides, or if we're building proteins, we need amino acids. And we'll talk about the rest of these things as we get into it. But it's first helpful to understand this, the genetic code. So what is that translation? So if we have that genetic information as nucleic acid, how do we go from that to amino acid? And so this is all about the genetic code and translating that. Now, DNA has 4 kinds of nucleotides, as we know, or we can say RNA nucleotides, just swap out T and U. But it has to code for 20 kinds of amino acids. So you couldn't just say, well, A stands for glycine, because you would run out of code words very quickly, right? The other question is, if we have this RNA sequence here, is it just read one after the other with no space? Or are there big jumps? Sometimes we jump letters? Do we skip them? Right? How do we figure that out? How can we know that? Well, there's some pretty simple experiments that can be done to figure this out. First of all, how many letters would you need to code for amino acids? If we say 1 letter at a time, that's not enough because you could only cover 4 amino acids. If we say a 2-letter code, that gives us, count them up here, 16, but still we have 20 amino acids. That's not enough. If we go with a 3-letter code, that gives us 64 possibilities, 64 combinations. And that's where we're headed. That would be enough. And then the question is, well, do all those code for something, or only some used? Again, do we know if there's spacing between them? Let's see what happens. And so there's some really nice experiments here by Crick and Brenner. This is Crick of Watson and Crick. And what they did was using directed mutation, deleting bases from a stretch of RNA. And so we see that here, if this is our RNA and here's a message, and here our presumptive code, what it codes for, our presumptive polypeptide, if we take away this one base, it will change the rest of this message from that point forward. So this suggests that there aren't just random spaces, because if you took out a base and it occurred in one of those spaces, it probably shouldn't affect the message. But they found whenever they took out a single base, it would always affect the message from that point forward. The other thing that's important, and it supports this idea that this information comes in units of 3, is when they would remove 3 nucleotides, shown here right next to one another, they would lose certainly an amino acid, but the rest stayed the same. So here's histidine, arginine, etc. Those were retained, whereas here the amino acids all change if we knock out one. And so this was the first ideas about how the genetic code works. And the code occurs in what are called codons. So these are sets of 3 nucleotides that specify an amino acid. And they are read continuously, so there are not spaces in between them. And it's also really important to understand what's known as the reading frame. Because if you just have a random string of nucleic acids, the message might be different if you start in a different spot. So let's say here's our reading frame, and we're just going to start at the beginning. Maybe there's more nucleic acids here, but we're just going to start here. If we start at this AUG, we get this amino acid sequence. But what if we started 2 in? We just skip those first two. We start here. We get a completely different amino acid sequence. So this is crucial. Knowing where to start is absolutely critical, because you get a different message if you start in a different place. And that's all about this reading frame idea. All right, so the stage is set now. How do we figure out which codon codes for which amino acid? And again, do some of them just not code for any amino acid? Well, this is how this was done, by using synthetic messenger RNA molecules and with predictable nucleotides. Say, for example, here's a string message that's just uracils, uracil, 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 etc. And what does that code for? Phenylalanine. So there you go. That means U, that codon, that is phenylalanine. Now you can do this with the other bases as well, just a string of repetitive bases, figure out what those are. You could alternate bases. So here's UC, UC, UC. That codes for serine and leucine, serine and leucine. Now the question is, well, which one is which? Which is UCU and which is CUC? That takes another experiment to figure out. But here we know that UCU, for example, is serine. And so by using this systematic process, people over a few years figured out which codon counts for which amino acid. And here's that genetic code. And there are several important things to notice about this genetic code. One is that there is a start codon. So here's this AUG. That is a start codon. Remember back to that idea of how do we know where to start reading the message? How do we know where to start translating? It starts at an AUG, a methionine start. So methionine is the amino acid. So every protein, at least initially, would have methionine, because that's the first amino acid that gets added. The other thing to notice here, look at these. Remember that question of, does every codon count for an amino acid? Not necessarily. So here's UAA, UAG, UGA. Those are stop codons. They don't have a corresponding amino acid. Another critical thing, the code is what's called degenerate. What does that mean? It means that you can have an amino acid coded for by more than one codon. So here, let's just pick at random here, valine. It can be coded for by GUU, GUC, GUA, or GUG. And it's usually that third position that is degenerate, meaning it doesn't matter what base is there. If you've got GU something, you get a valine. And we could just use another example. Here's alanine. GC something equals alanine. And so this is called degeneracy. And again, often it occurs at that third position. All right, so if we have a long random sequence of RNA, how often will a stop codon appear? All right, yeah, this is just a basic statistical question. If there are 3 stop codons and 64 codons total, then 3 times out of 64 at random is how often that's going to occur. Okay, here's your chance to decipher the genetic code. Here is a piece of messenger RNA sequence written 5' to 3'. And what will this code for? And I give you a little bit of a hint here. Make sure you start in the right place. Okay, so, well, right away, if you know where to start, which is the start codon, you're looking for AUG, and the first one we see is right here. So we can spread this sequence out by groups of 3. That'll help you translate it. And really, only one that even starts with methionine is B, but the rest of the codons match up for these amino acids. And notice isoleucine AUU as our last one here. This UAG is a stop codon. You could write stop, or you could just end the sequence there. That's actually what happens, right? The protein doesn't have the word stop attached to the end. It just ends at this amino acid. So let's get back to that central dogma now that we understand a bit about the genetic code. And we'll start with transcription. So this is the first step. And there are some big sort of questions or how we think about transcription. How does it differ between prokaryotes and eukaryotes? Where does it begin, right? It has to start somewhere. And because DNA is double-stranded, which strand of that double helix actually gets copied? And we can split transcription into 3 phases. And we saw these phases with DNA replication too, and they have the same logic behind them. It has to start, so that's initiation. It has to happen, and that's called elongation. What are we elongating? A piece of RNA, messenger RNA. And then it has to end at some point, so that's called termination. So it has to stop somewhere. And we have to remind ourselves during elongation here that we're going to be building our piece of messenger RNA 5' to 3'. Just like when we saw when DNA was being copied, our new strand was complementary and anti-parallel. We'll see the same thing with the messenger RNA. And so we can look at that as an example here. So here are 2 strands of DNA, 5' to 3', and anti-parallel strand below. If we separate these and we're going to make a piece of messenger RNA copy, which way would each of those go? So on this top strand, would it grow from left to right or right to left? Remember, it's got to be anti-parallel. All right, it would grow from right to left because here it's anti-parallel. And then we can do the same with the other strand. It would grow the opposite direction or be synthesized in the opposite direction, anti-parallel. And so that is going to be critical. We always see these anti-parallel interactions. If you don't have a handle on this yet from the previous chapter about DNA replication, this is time to really grasp it because we're seeing it here again. So the next question is, well, which DNA strand gets copied? Which one gets copied, or which one is probably a better way to put this? Which one acts as the template? And we can just use this example here. Here's our double-stranded DNA, and we're separating it. In this case, this bottom strand is acting as the template. And so what do we see? If it's 5' to 3', right to left, our piece of message is being built from left to right, anti-parallel. And base pairing here, complementary base pairing, A's go with T's, U's go with A's, etc. This other strand, the non-template strand, it's just kind of pushed out of the way for now while this piece of message is being made. It'll come back. But a handy thing to notice, this is sometimes also called the coding strand. That can be confusing sometimes because sometimes when you're learning about this, you think, well, oh, that's the strand that's copied or used as the template. But it is the non-template strand. When in doubt, go to the template strand and just start base pairing stuff. But as you get familiar with this, you can see a shortcut here. Your coding strand, look at the sequence, C, G, A, T, and look at our piece of RNA, C, G, A, U. This is why it's called the coding strand, because if you were to read this, even though ultimately this one is just temporarily shoved out of the way, it has the same sequence, except T's being replaced with U's, as the piece of message that's being built. All right, now you try. So here's a DNA sequence. You've got your orientation. You know which is template, which is coding. Given your choices, which piece of RNA will be produced from this? All right, so knowing what our template strand is, we'll just shove the coding strand out of the way and base pair our piece of message anti-parallel to our template. So it has to be going the opposite direction. And so this would be U, C, G, G, etc. Just pay attention to the orientation here. Again, or you can use that shortcut knowing that the coding strand would have the same sequence except for the T's being replaced with U's. All right, now that you have the relative orientation of these different nucleic acid strands down, let's actually look at what happens. So transcription starts with initiation, and the molecule, or the enzyme rather, that actually carries out the copying of the DNA or producing that piece of RNA is called RNA polymerase. Very fitting name. So it's going to make a polymer of RNA. And as we already know, it only can grow in a 5' to 3' direction. And then the question is, like, how does RNA polymerase know where to start? It doesn't need a primer like we saw with DNA polymerases back in DNA replication, but it needs to know where to start. And so what is that? It helps to look at the transcription unit here. At the start of a transcription unit is something called a promoter. And this is a region of DNA, as we'll see, and the sequence determines this, where RNA polymerase knows where to start. It's actually other proteins that bind there first, as we'll see, and then RNA polymerase picks up from that point. And so our transcription unit starts with the promoter, then we have the coding sequence in the DNA here, and then ultimately it'll reach a stopping point, that's the terminator. And so here we can just have the big broad view of transcription at this point. Here proteins bind to the promoter, RNA polymerase comes along, it binds, it starts the RNA transcript, and it opens up, sort of unwinding this DNA, creating this replication bubble, or transcription bubble, I should say, in this case, transcription bubble, and producing this complementary strand to the template strand, and ultimately hits that terminator, and off comes RNA polymerase. But let's go back to the promoter, because we're still talking about where do we start. And so in eukaryotes, for example, the eukaryotic promoter contains this TA-TA sequence, and this is called the Tata box. And you'll notice here on our figure, we have the non-template and template strand. That TA-TA, that's actually on the non-template strand. So the non-template strand is acting really as the binding location for those initial transcription factors. Remember, I mentioned it's not RNA polymerase at first. It's a few other proteins that will bind there, saying, hey, this is a gene that needs to be turned on, and they will attach at the promoter at that Tata box, and then that acts as a place that RNA polymerase can now bind. When all this comes together, we've got the promoter, we got our transcription factors, RNA polymerase comes in. Now everything's together, and this is known as the transcription initiation complex. And off goes RNA polymerase. Those other proteins get left behind. Now RNA polymerase makes that RNA transcript. And that is through what we call elongation. So this is that next step or next phase in transcription. And so we see that transcription bubble open up. The DNA unwinds just temporarily, about 10 to 20 nucleotides at a time. And another interesting thing that can be happening is those transcription factors that were left back at the promoter, they are now still there in sometimes, and a new RNA polymerase can come along. So while one is zipping down the gene here, making an RNA copy, another one can be attaching, and then another one can be attaching, etc. All right, so how quick is this? It's pretty quick, 40 to 50 nucleotides per second that RNA polymerase can zip along. So these genes can be transcribed fairly fast. So how does RNA polymerase, quote unquote, know where to stop? Why does it stop? Well, it reaches a sequence of DNA that is really a signal for it to stop. And why does that work? Well, it differs between eukaryotes and prokaryotes. And here's an example of how this could work. So RNA polymerase will reach a certain DNA sequence. What you see here is a GC-rich area, which will form a hairpin in this new piece of messenger RNA. And then RNA polymerase will hit a bunch of adenines, and therefore it's plugging in uracils to complement it. And this sort of slows it down. And the combination of this hairpin and these weaker AU bonds will result in the transcript being yanked out of the polymerase and terminating it. So it's all about the DNA sequence and the properties of it that determine that terminator sequence. At this level, what's important to know is that RNA polymerase hits a terminator sequence, and that's why it stops. Now, what happens to that transcript? We'll see there's some differences between eukaryotes and prokaryotes here as well. That piece of message in prokaryotes can just be translated, but in eukaryotes, it needs to be modified. And what we will see is something called a polyadenylation sequence near the terminator, and we'll talk about what that is shortly. Okay, let's put you to the test now. Here we have a couple of strands of DNA separated, opened up. We have a place where there's a promoter and the orientation of the promoter. And notice the orientation of these strands, 5' to 3', on our DNA strands. Which of these letters in the orange, these are potential RNA transcripts, which of these could actually represent a true transcript? Okay, yes, only Y. I mean, first of all, you know the promoter is oriented to the right, so it could be Y or Z, but then it comes down to the polarity of your strands. So if our RNA is going to be anti-parallel to the DNA, this is the only one that works because it would be 5 to 3', anti-parallel to our DNA strand, which is going 5 to 3 from right to left. Z does not work because this would not produce an anti-parallel strand. All right, so remember, in prokaryotes, you've produced that piece of message. It can be translated right away. So even as that piece of message is being produced, coming right off of the genome, ribosomes, those protein building machines, they start attaching to it right away. And we can see that here where we've got, here's a genome here. So here's DNA. You can barely see it here, or you can look at the cartoon. And coming off it here are RNA strands. Notice they're longer. The further you get along, the longer the piece of message is going to be. But right away, ribosomes are attaching right onto this and producing, here we see the little proteins actually being built as this piece of message is being translated. And we'll see something similar later with eukaryotes once the message is processed and it's elsewhere in the cell. That they can have multiple ribosomes attached to a single message, and these are called polyribosomes. But this coupling of transcription and translation happening all at once cannot occur in eukaryotes because we have the different locations where these things occur. Transcription occurs in the nucleus, RNA needs to be processed, and then the message can be translated out in the cytoplasm. So let's talk about that RNA modification or processing. There's a few things that happen. Well, one, we get a poly A tail. I mentioned this when I talked about where RNA polymerase terminates, near this polyadenylation signal, a little ways after it. After our piece of message is produced, this signal acts as a location or marker for another enzyme to add this tail of many adenines, so poly A. It also gets a 5' cap, so this is actually a modified guanine with 3 phosphates added to the end of it. Now, what these are important for is providing stability for this piece of message. There are enzymes that would chew away at the message, say from this 3' end. By having a tail on it, it takes longer for those enzymes to get to the point where they're actually chewing away at the coding information. And then similarly, this 5' cap provides some stability for this transcript. This might also be important for getting that piece of message to the place that it needs to go as well. Something else that happens with our eukaryotic message is that our genes, eukaryotic genes, are not just continuous coding sequence. They have two components to them, what are called introns and exons. The exons are the coding sequence or the expressed sequence, hence the name exon. The non-coding regions are introns, the intervening sequence. That's why they're called introns. And so for this message to be read correctly, the introns have to be spliced out. So the whole transcript is made from this gene that contains introns, and then the introns get cut out. And you can think of it like this. Here's an example. If we had a message in English where we had interspersed letters in amongst the actual message, we can't really read the message until we remove those interspersed parts. So here we have introns, we cut them out. Now we see the actual message, and it's the same here. So here's our pre-RNA, and introns are spliced out, and then we have our final transcript. Another really cool thing about this splicing is that you can have alternate RNA splicing. So here's our gene, and we have these exons, 1, 2, 3, 4, 5, and then introns in between them. And this is an actual gene. This is the troponin T gene. Here's our primary transcript. Again, it's got everything, introns, all the exons. What can happen is not only the intron spliced out, but sometimes an exon is spliced out. Actually, I should say all the time an exon spliced out in this particular example. Sometimes it's the number 4 exon, and that produces this piece of transcript. Sometimes it's the number 3 exon, not number 4, and it produces this transcript. So from a single gene, you can actually make 2 different things. Now, there are a lot of similarities between these two because they share exons, but there are differences. And so this is really important when we think about genetic diversity, how that equates to phenotypic diversity, because in this case, one gene actually equaled 2 different transcripts. So what does all this splicing? It's a structure called the spliceosome. Pretty clever name, right? Very fitting. And this is what will cleave out those introns. And again, sometimes exons go as well, as we just saw in that alternate splicing example. But this highlights this point I've made a few times, the idea that not all enzymes are proteins. A spliceosome is made up of protein and little bits of RNA. And it's actually the RNA that will bind to the intron RNA. So we see that here. If you zoom in, you've got the RNA in the spliceosome binding to the intron RNA in a couple of places. And it's the RNA itself that's the enzyme here that cuts out the introns. And so when you have a catalytic RNA, they're called ribozymes. And we'll see this is a recurring theme when we look at how gene expression works. And so the spliceosome is just one example of these catalytic RNAs, and it's very important for cutting out those introns. Interestingly enough, some introns can actually catalyze their own splicing. So that intron sequence itself is a ribosome, and it will cut itself out, which is totally awesome. All right, we'll tune in for the next lecture. We'll talk about how this message is now translated.